I just want to begin by introducing our chair for tonight's event, Prashant Shah. He's the CEO of the O2H Group and O2H Co-work Labs. So just a brief introduction to both the O2H Group as well as Prashant. The O2H Group has a vision of seeding new ideas in life sciences and AI technology, green innovation. The divisions include funding, incubation, and execution, covering chemistry, biology, and technology. The group is headquartered here in Cambridge in the United Kingdom and has nurtured an east to west footprint encompassing the United Kingdom, the USA, and India. O2H Co-work Labs also operates from the Mill SciTech Park, which is shaping an experience sharing community across techies and scientists and CXOs, VCs, academia, and the big CEOs. So Prashant is a serial entrepreneur in life sciences and technology, in which one of those companies was acquired by a public company. He's currently active in seed investing, product and IP development, services, and then also now as well building lab and office infrastructure. So his earlier career was with the strategy group at Accenture, and he has a long history here in Cambridge, actually getting his MPhil in management from the Judge Business School. So welcome to you, Prashant, and welcome to all of the other panelists in this evening. Over to you, Prashant.
Thank you very much, Matthew, but also to Olivia and Claire and Rebecca and the whole kind of Cambridge Enterprise Tuesday team. We're doing a wonderful job of getting the message of entrepreneurship and science and innovation out there into the kind of the Cambridge world. You're a pleasure to work with, and thank you for the opportunity to kind of to host this absolutely fascinating topic, genomics and AI. It's kind of absolutely mind-boggling. And you know, I've had the opportunity to kind of get to know our three panelists, John and Louwai and Debora, quite recently. And the more I talk to them, I think that adage goes, the less I realize I actually know. They're an absolutely incredible bunch of people we've got on this session. They're genuine and real pioneers in what they're doing, and they're all covering different spaces in what we want to talk about today. So I think it'd be a good opportunity for me to kind of just allow them to introduce themselves. John, would you like to go first?
That's better. So thanks, Prashant. My name is John Moore. I'm a biochemist and cell biologist by training. I've been in industry 19 years now, first of all with an analyst in small molecule drug discovery, moving to be CSO at Horizon Discovery, evangelizing gene editing as a target validation tool. Subsequent to that, I became an operating partner at Advent Life Sciences, where I man startups basically. I have a new one starting this week called Epitopia, which I'll talk about more. And I know Prashant, and particularly his brother, through being executive chair of Pencil Biosciences, which is a 12% FTE gig for me, which is developing an entirely synthetic genome editing tool from scratch. So it's designed rather than discovered, invented rather than borrowed from Phage. So it's quite novel. We're excited for that. So it's a pleasure to be here, and I'll let someone else speak now.
Louwai, would you like to go next?
Sure, thanks. So my name is Louwai. I'm a medical doctor by background. I'm a practicing neurologist, trained in the UK, and been working in London most recently. And my other background in academia is in genomic medicine generally. So I've been working in Professor Johnson's lab at Imperial around that area for a little bit of time. And I've been part of this spin-out company from Imperial called CoSyne Therapeutics, where we're sort of developing some new technologies and new techniques to build the next generation of genomic precision medicines.
Debora.
Hello, hi, my name is Debora Lucarelli, and I'm an Genomics CEO. I also come from a technical background. So after completing my studies, I had the pleasure, the opportunity to join various ventures such as Oxford Nanopore Technologies and Cambridge Epigenetics, and work on very new and cutting edge platforms. I've also had my own labs within the University of Cambridge, where we were beta tester for many new platforms. So about a couple of years ago, I had the pleasure to meet Professor Frasers and the other co-founders of the company, and we decided actually to start our own ventures, hence Enhanced Genomic was born. Pleasure to be here.
Thank you. Thank you all of you for your elegant introductions. And I'd say my own brush with kind of the world of genomics went back to kind of 1995, where I had an opportunity to work with John Sulston. He's a Nobel Prize winner. He was working at the Sanger Center, and we were in the thrust of kind of sequencing the human genome. And I was, one of my tasks was to, as part of my thesis, was to kind of see if I could help speed up the PCR reactions and the overall kind of sequencing timeline. So that was my kind of early kind of contact with the world of genomics, and it's been a fascinating story since. And I'd like to perhaps start with yourself, John, and kind of just, if you could kind of go into your current research area or recent research area in terms of gene editing. Now, it's something that is quite an area which kind of fascinates and intrigues in many ways. So if you could kind of tell us about the current gene editing technologies which are out there, and perhaps, you know, where your research area has been and what you're trying to do that's different in terms of taking it forward.
Yeah, sure. So gene editing, really, you have to go back about 40 years for the origins of it. I remember having a dinner when I was a postdoc at UNC in America with Joe Heitman, who we had some visiting speaker, and he just came out of blue. Wouldn't it be great if you could engineer a restriction enzyme to be completely specific for you know, cutting individual regions of the human genome? And so this is back in 95, 96, total pipe dream at the time. Then there were some very, very specific nucleases around for the biologists in the audience, which is hopefully most people. They know what a restriction enzyme is, and there are specific examples, but very, very difficult to alter to get the function you want. The first big step forward came in Cambridge, actually, with a now largely forgotten company called GeneDAC, which had some connection with Aaron Krug at the LMB at the time. And this was taking naturally occurring zinc fingers and modifying them so they could start to recognize triplets of DNA. So you could build up a sort of modular restriction enzyme which cut DNA and bound specifically based on these, I think, fairly complex and difficult to design and a hard protein engineering project to get it to work. So they ended up reaching the clinic at a company called Sangamo from about 2010 onwards. So this was a slow builder. There were things involved in research tools. Sigma sold them. It was all 10,000 quid a pop, probably £1 million if you can be treated with it, and pretty slow. Some other technologies came along, Talons, which came out of plant transcription factors, and a company called Selectis was developing those, and they switched into sort of CAR-T type therapeutics. And then when it comes to gene editing technologies, the big advance really, which has got it in all the newspapers and now has every undergraduate wanting to do it, came in 2012, where Charpentier, Doudna, Keith Young, and Feng Zhang and other people managed to get this obscure sort of phage-adapted gene editing system, which had been shown to work in bacteria earlier in the year, to work in mammalian cells. Now, this was something people noticed. And the key thing about CRISPR-Cas9 was it was child's play to program it to cut a piece of the genome one wanted to, because all you needed was 20 nucleotides of RNA. So that democratized it as a lab tool, you know, which has led to the Nobel Prize. And it led to the launch of another load of aspirational gene editing companies. CRISPR Therapeutics, Editas, and Intellia have been the first wave, and there's been others since, to go after the applications of this in the clinic. And you know, there are a number of ones. You can there are a number of diseases one can go after gene editing. I'll go through a few of them, which are realistic. So it's most easy to take cells out the body, fiddle with them, see what you've done, and put them back. And this is a realistic sort of approach to treating sickle cell anemia, beta thalassemia, by fiddling the hemoglobin locus. There's various ways you can do it, and lots of companies are trying. When it comes to edited cell therapies, there's a big interest in CAR-Ts, TCR-Ts, etc., etc. These tend not to persist in the body or have to be made anew for everyone. And there's the potential of coming out of off-the-shelf, there'll still be expensive products, by editing donor cells so that they no longer attack the new host and are not recognized as foreign by the host either. And there's also some technical innovations one could do about persistence as well. So these are the two main editor areas where gene editing, there's sort of parallel projects going on. But the technology also has the promise of treating cells in the body. And there's two examples of this being tried in the clinic with CRISPR. So Editas are introducing the system with viruses into the eye to go after various sort of retinal problems. And most recently, Intellia, using lipid nanoparticles similar to the COVID vaccines, have been able to knock out genes in the liver and reduce the production of toxic metabolites. What Pencil is doing is I suppose, can crudely be characterized as an IP bypass. So the patent situation with CRISPR-Cas9 is extremely complicated. Different patents look like they're going to survive in different territories. There's a whole host of alternatives to Cas9 coming along in the background with unclear IP. And the founder of Pencil, who wasn't me, a guy called Raja Ibrahim, who was working in Singapore at the time, he used his background to look at, stripping it, this sort of CRISPR-Cas9, which is 4,500 amino acids, down to its absolute, nucleotides, 1,500 amino acids, down to its absolute basics. So he designed on a piece of paper a nuclease domain. He looked carefully at how CRISPR-Cas9 worked and designed a very sort of mini version of it, completely from scratch, with fully synthetics, either designed or selected components. And just in the past couple of months, still a stealth company, we've got evidence of gene editing at a reasonable level in mammalian cells. And so that puts us potentially in a position to do the wonderful, weird, and interesting things to the genome that one can do at CRISPR-Cas9. That's a company at Audley Park. We have an endless list of things we could do, most of which would be a fatal mistake. And we have to put our thinking caps on, work out what the technology is best for in terms of differentiation, and what opportunities still remain, which aren't completely, all the patients lined up for clinical trials with other agents, etc. So we've got a lot of thinking to do about how to bring this technology into the clinic. And really, that's where the only place one can go. I think when it comes to lab tools, there's no point in another CRISPR. Scientists are innovative, but also conservative about the tools they use. And so we see the therapeutic applications as the only serious place to go with this technology.
Thank you, John. That was a great tour of what's happening in terms of gene editing, in terms of CRISPR, Cas9 technologies, and even your own innovation as well, and what you're working on. Louwai, you're kind of looking at some very interesting areas, and also looking at where kind of machine learning can kind of support drug discovery and understanding the world of understanding the human genome. So would you like to kind of jump in and just explain to us your kind of research area and how that's translated into a company?
Yeah, sure. So origin of CoSyne Therapeutics, it really grew organically from academia, and it came about from the shared passion we all have about translating academic science into something that can help patients in the real world. So Lisi, my co-founder and scientific chief science officer, she had been working in cancer for many years and had a very strong expertise in brain cancer particularly. And my other co-founder, Professor Michael Johnson, had been working on drug target discovery and genomic systems biology in neurology and neuroscience for a long time. And no one had ever really applied the tools that Professor Johnson was using in the neurology sphere to the cancer sphere. And so when Lisi joined to do a PhD in his lab, it was a great cross interdisciplinary collaboration to try and develop that and apply those resources and those tools in a new setting. And often that's when some of the best innovations happen, and you take these sort of ways of thinking from one sphere and apply it to another sphere. And what we have been developing is effectively a computational engine that allows us to identify precision medicine drug targets in a way that's completely novel. And it utilizes a part of biology that allows us to go and target what's prevented previous precision therapies from succeeding. So in particular, a lot of cancers are molecularly heterogeneous. And what that means is within any sort of cancer tumor, if you start biopsying from different places, you'll find different populations of cells, different molecular features, different genetic vulnerabilities. And if you create a precision therapeutic that targets a subset of those cells, then you're always going to drive an evolution of resistance, and you'll have a tumor recurrence, which is now no longer susceptible to your drug. But what we've done is being able to identify tumor biomarkers that occur very early on in the evolution of a cancer. So these are features, genetic features that are conserved across all the cancer cells in the population. And we're able to, using this process of synthetic lethality, turn those molecular genetic features that are highly conserved into vulnerabilities that we can then exploit with precision therapeutic drugs. So that's the basis of our computational engine. But what's exciting about our engine is it's actually very widely applicable. We're using it in cancer we're applying these machine learning approaches to identify features and extract features that predict why cells will die in certain situations. You can actually reverse that to predict why cells will survive as well. So you can imagine applying that same type of engine to a neurodegenerative process and solving a feature selection problem whereby you're no longer identifying what makes cancer cells die selectively, but what makes neurodegenerative cells survive. And so you can then start applying the same engine to other areas of neurology as well. So it's a really exciting field that allows you to bring in these two aspects of genomic medicine and AI at the same time to achieve quite a unique goal.
And why did you choose to kind of explore kind of glioblastoma first?
So that's to do with the fact, I think, there's two reasons. One is our expertise and background has largely been in neuro-oncology, but secondly, these are really devastating diseases with massive unmet needs. The current gold standard of care in glioblastoma, temozolomide, prolongs life expectancy of about 3 months. And that's pretty rubbish since there have been no new drugs developed since 2005. And so part of coming at this problem as clinicians means we interact with these patients, we know them. So I often see patients with these cancers presenting with seizures, and we deal with these patients on a day-to-day basis. We have relationships with them, we understand what matters to them, what they want to do. And seeing that on a day-to-day basis really changes your perception of science. It's no longer you sitting in an ivory tower thinking broadly about cool different parts of science. You really can see how this translates to the person in front of you and how to help them. And we saw this as a huge unmet need in this area, and we thought that this would be the best place to try and apply this technology to get treatments out to those patients as quick as we could.
Thank you, Louwai. Debora, you've got some working in the broad field of kind of epigenetics, and you're looking at non-coding DNA. Would love to hear a little bit more about it. Please,
yeah, that's
tell
right.
us.
So like Louwai, actually, the founders of the company have been working in regulatory genetic regulations for the last 20-25 years. So they have a lot of experience in, exploring the human genome in generally, and they have developed many famous platforms. So they worked a lot in IC. There were some of the early users, and also the first to map single cell IC. They've done also other type of discoveries. So when they developed within their academic setting this promoter capture IC, is because they've started thinking, hey, how about we're trying to have a real view of what's happening in a cell in one single experiment. And the idea was, how can we target every single gene? And at the same time, how can we actually look at what's happening, in the rest of the genome? And that's how they came up with this platform that allows you actually to detect every single gene in one single experiment and get configuration or information about the interaction of every single promoter. Now, we always say we work in the space of 3D genomics, and we really think this is, the next generation of genomics in itself. We all used to think of the genome as that 2D or the long strings of DNA, and we have 3 billion of base pairs on it. And there are lots of tools actually to date to identify every single one of those. But if you think about it, at the moment we use only 2% of that genome, because only 2% codes. For proteins that they are actionable, we can design drug targets. And on top of this, so there is this 98% of the genome that needs to be explored. And we do know that 95% of the mutations, actually, of the single nucleotide polymorphism, single point mutation that actually are responsible for disease are actually located in the non-coding genome. So there is a vast resource there that's been really unexplored, because all those mutations are sitting there, and we need to try to work out, how are they affecting, genes. So we've developed this platform that allows us to systematically understand, how these single point mutations are connected and which genes are they affected by. And we've been starting this by building initially a database and then comparing actually the profile across different types of cells. And then by doing that, the founders of the company have noticed, hey, we can see now that actually people have different genetic profiles, and we can see actually that there is a different response. And that's when we really thought, I think here a commercial entity will be, better place to do a more systematic discovery and start looking in drug targets, or, on targets for biomarker or epigenetic marker. And so that's how we started our venture, actually. So do you have a question, Prashant? I can see you are about to.
I'm about to. You're quite right. So, I mean, you seem to be kind of plotting the future of precision medicine here, really, with your understanding of the non-coded part of the DNA.
Yeah, and I think in many ways we all are here. So for each of us here with his own different stories, both myself, John, and Louwai, actually, I think we all are thinking that this is where, the future is. So the concept here, like, I think we all use this, similar concept, actually. We need to collect some data, like Louwai previously announced, it's important to kind of train your algorithm and your machines so that you can start developing some predictions, and you can start also understanding specific patterns. And I also tell everybody that what I'm building, I'm telling this to my investors, and every time, Peter, we are actually building what we would like to call an artificial labs. So we think that by collecting enough data, by real data, and by profiling actually a lot of patients, we will be able in time and in the future actually to start thinking of more precision medicine approach, actually. So this is actually the aim of the company. And if I understood correctly, I think also the other speakers believe into this kind of philosophy, let's call it like that.
John, we were speaking about precision medicine earlier. Where do you think I mean, you've got, firstly, would you like to elaborate on a couple of, you've got a portfolio career, actually. You're kind of a CEO of a couple of different companies. You're a chair of a pencil as well.
I don't recommend it, really. Of being a parallel entrepreneur, it does have limitations in focus.
You're a triple entrepreneur, at least. I mean, I was kind of working through it all. It's quite
I'm trying to simplify my life a bit. So I think I had some very relevant experience when I was at Horizon, where I think back then we were thinking of doing this around 2016, 2017. And first of all, we didn't have any informaticians in the company. And I think the data sets to train algorithms were to look at important nodes, etc., weren't out there. So we did try to do some cancer target ID, not in glioblastoma, and we worked in colon cancer in particular, because that was a strength of Horizon at the time. And so we did some CRISPR screens, purely empirical ones, just in cell line models, but we focused on relevant genes, which not for understanding control, but for actually coming up with actionable targets. We probably should have focused more on antibody ones, but we included small molecule targets as a rule. we found some specific vulnerabilities, and we're able to validate them within our own set and a somewhat wider set of cells, and they were extremely convincing. We didn't just tag the gene as important, we tagged individual and functional domains as important. Discovered some vulnerabilities of KRAS cancer, tried to set up a spin-off company around this, which is how I ended up working in venture capital firms. But there wasn't sufficient data to be funded there with the story we had. So those targets ended up being partnered with public company C4X Discovery, which really could do with more money to exploit them. And I moved on. Now, with Pencil, to return to that sort of technology is best for going after, clear Mendelian inherited, problems that patients have. And this is, if you've got a gene, if you've got toxic metabolites, there might be an enzyme you can knock out, which might not be affected in disease, but it's something that is dispensable, but its elimination removes the toxic metabolite. So that's more or less what Intellia are doing. You can look at it as a sort of synthetic viability screen on the level of the body if you want. But it's something where if you can do it, if you've got that penetrance of the effect in the liver, you'll rescue the patient and the disease will stop getting worse. And similarly, in cases with beta globin issues in adults, where the hemoglobin is defective or absent or lower expression, it has been shown with small molecules as crude as hydroxyurea that if you muck about with the epigenome, you can get fetal hemoglobin expressed. And therefore, by targeting the genetic elements that keep that suppressed in adults, which is called the BCL11 gene, you can boost, re-express fetal hemoglobin and rescue the disease. And that you only have to treat the blood lineage for that to work. Job done. And so CRISPR Therapeutics has done this with a couple of patients, and they're doing fine. So that's very, very precise medicine. It's directly at a particular Mendelian defect, and if you can do it, it will work. The other thing we're doing at the moment in the new company Epitopia is to try and come up with an effective cancer vaccine. So there's this new class of tumor-specific antigens, which are encoded by the dark genome, if you like. So you could say they're caused by dysregulated transcription in cancer or dysregulated splicing. And all of those things can make major differences in the protein content of a cell. And some fraction of that will end up being presented on the MHC system and therefore be available for T cells to recognize. So it's by using mass spec, an empirical technique, that this sort of rich potential source of antigens has become apparent. So then they're not something anyone's found by looking primarily at sequencing data, whether it be RNA or DNA, or 3D sort of analysis of the epigenome or anything like that. It's been done by a relatively old technique, if now done with modern instrumentation, to find these potentially tumor-specific antigens. But there was a big transcriptomic component, I mean, in understanding which of these peptides, which may or may not be there, because it's a very, very error-prone process, mass spec, were encoded by cancer-specific transcripts. So that's the insight of our founders who are in Canada. And so we think that represents 85 to 90% of at least the peptide components visible by T cells. And so therefore the major opportunity for cancer vaccines.
Great, thank you very much. I'd like to just kind of look across the kind of the various gene therapies that exist out there. We talked about gene editing and even kind of the vector-based therapies. Louwai, would you like to kind of give your views on some of the gene therapies that exist and which ones are work and what you think the future holds for some of those?
Sure, yes. So I guess the first distinction, if you're talking about gene therapies, is whether you're talking about somatic gene therapies or germline gene therapies. So there's various reasons why both ethically and technically they're very different. So I think with somatic gene therapies, you're talking about either your standard in vivo introducing a gene modification via a viral vector or some kind of plasmid or nanoparticle or an ex vivo approach like we talked about, where you take patient cells, modify them outside, and then reimplant them back in with the genes modified, so CAR T therapies. And those are really quite exciting and cool technologies. You used to watch 1980s films where people have robots that get miniaturized and then swim around the body and heal things. It's basically what CAR T therapies are. You're taking robots, your T cells, reprogramming them and putting them back in to achieve a goal. So those sort of technologies, they're really a lot of potential, a lot of excitement that can be done there. And there's a lot of things that you could change. In the neuroscience sphere, which is where most of my expertise is going to be, I think because of problems with getting the genetic modifications to the targets, blood-brain barrier penetrance, there's a thick skull in the way. I have a feeling that people are going to look at the way we did that as quite barbaric. So right now what we do is we have to cut out a flap of the person's skull and then basically injecting a virus into a bit of brain. And I think that's going to be in the future seen as a very primitive way of doing things, but it's currently the best thing we've got. And eventually I think there'll be opportunities to overcome that. But the somatic technologies are all effectively just technical changes. There are a new way to achieve the same goal as any other sort of medicine would do. But with the germline mutations, it opens up a whole different type of issues. So germline genetic therapies, whereby you're actually going in and altering the genome that will be inherited across the population. So from either an embryo, changing that, or changing the sort of gametes that are inherited. You're now opening up lots of problems with ethical considerations, consent about the patient that you're treating, what's the fine line between enhancement versus therapy. And there's also aspects to some of the more advanced, I guess, gene therapy type ideas, things like gene drives, where you're basically putting in the CRISPR apparatus into the genome as well as the gene that you're trying to edit. So instead of having, say, a gene inherited in a Mendelian fashion, you have 100% inheritance of that gene because the CRISPR system puts it back into both chromosomes. And so within a few different generations, the entire population has been edited. So you're now not even talking about editing individuals, you're talking about editing populations and entire ecosystems. So there's a massive amount of potential in that technology. So if you were to edit in malaria resistance into mosquitoes, and there's a huge amount, it would be unethical to not save the millions of people that are dying every year with that disease. But at the same time, if you're engineering the female Anopheles mosquito to die out, and now the population becomes all male, and then the whole population of mosquitoes crashes, you have no idea what the consequence of that will be to some crop production in some part of the world that maybe relied on 10 different orders of magnitude changes of things that occur. So it's a very complicated field, and I think the world is still a bit far away from really grappling with that when it comes to germline editing techniques. There's the recent controversies in China with Dr. He's lab, where they edited those two girls with HIV resistance who were born human edited, Nunu and I think they were names. So it's really a lot of exciting potential there, but obviously with any big technologies, there's a lot of things to consider as well at the same time.
What's the kind of status of regulation around some of these issues that we've touched upon just here? Is the regulation robust enough to protect us against some of the risks from people kind of using these powerful technologies perhaps in the wrong way?
So it goes back to, I think, the comment about democratization. So before, this was a very difficult thing to do, and it was basically regulated by the fact that there weren't many people that could do it, and scientists tend to regulate themselves at that sort of high level. But now when you've got it being so simple, as it was mentioned, it's effectively child's play to just change some of these things. You're now always going to have rogue scientists and rogue groups who are exploiting vulnerable patients that are desperate for certain treatments. And I think it was interesting to see what happened with that China case study with Lulu and Nana, I think the names of the two girls who were born. The community all sort of came together to point out that really wasn't acceptable. We're not at the stage where A, the technology is ready to do that, and B, the implications have been thought through well enough. So science have often been quite self-regulated to that extent. But then there's also the questions about whether there should be some kind of overarching regulatory body that would come into play to oversee that, or whether governments should be regulated at that level, and different societies have different values. So it becomes a global question rather than something that you can regulate in a more traditional sense. So currently there are lots of ideas. I think when that was published, Feng Zhang's group, who are behind a lot of the CRISPR work, declared they sort of announced they wanted a moratorium. Many groups came together saying we just want to pause on this technology for the time being until we've worked out really where this is going and what we should be doing with this. And that's not the same as saying we should stop it, because I think it would be unethical to not intervene and treat a lot of these diseases with this type of technology if we have the ability to do so. But the first part of the Hippocratic Oath, and the most important part, is first do no harm. So I think before we get to that stage, we have to be sure that's what we're doing. And then we can look into how to apply these technologies to solve those problems, but with a first do no harm principle at heart.
Fine. So I'd like to, John, Debora, would you like to kind of chime in on these kind of gene therapy technologies and some of the implications of them?
So I haven't thought about this for quite a while, which may surprise you. And that's effectively because what was commercial was already clear. Now, in certain jurisdictions, you can get funding to make super pigs and this sort of stuff. And it just doesn't seem like a viable way forward. You know, in terms of what I know about autoimmunity, that the fact you're going to do 1,000 gene edits on pigs and come up with easy replaceable heart valves, which won't be rejected, just seems like a fantasy to me. So I haven't taken any notice of this. I know that if you are a company that licenses the CRISPR technology from the Broad Institute, the license has all sorts of restrictions on germline editing. And I think there's a number of countries have banned it and a number haven't. It's difficult to get consensus on. I think this is a technology which is an ethical rather than a danger in terms of, risks of things going wrong, so to speak. If you want a technology where things can go wrong, there are thousands of people who've got the sophistication, to make RNA and make any flu virus they want. With COVID, it's probably even easier if you wanted to make highly pathogenic ones. You don't need any gene editing to do that. It's just basic molecular biology and making things. Don't try this at home, kids, by the way. But there's a lot of technologies which are a realistic risk to the economy and health. Gene editing isn't one. I think it's an ethical point about what is human, what are scientists and doctors trying to do, and what are the commercial imperatives, which aren't that important, because there is no money in there's not hundreds of thousands of pounds to go in treating an embryo. And the technology probably would work, but the trouble is you don't know what else you're going to do in that situation, given the complexities of epigenomics and the lack of clarity on catalyzation and development if someone's messed with the genome. So I don't think anyone's going to go there, except in a sort of Dr. Moreau type way. So it's more like left field nutters, trying to make a name in weird circles. I mean, there are technologies, which have big risks of doing harm, which the world may bring into practice, like removing carbon from the atmosphere, seeding oceans of iron, blah, 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 which have unpredictable consequences. If you're looking at malaria, I mean, there's a problem. It goes up and down with, insecticide use and other things, but the world lives with it. Populations in malaria-affected countries still increase. There's obviously a big selection for heterozygotes with sickle cell anemia genes, etc. But when it comes to technologies which the world needs to survive, there's going to be a bigger issue of regulation and control, because there you have some societies will benefit for something and some will definitely be harmed in unpredictable ways. But I don't see the sort of genome editing technologies as at the forefront of problem causing, though they are, I suppose, at the forefront of ethics.
Debora, did you want to jump in? Yeah.
Yeah. So I presume I'd like to just take us a level up. Actually, I was thinking, I was just rereading the advertise for today's talk, and I presume we are talking so much about the gene editing because the topic was genomics. From a therapeutic and very elegant view, people immediately assume that you do, genomics discovery. You have, some genomics information, and the most obvious tool tend to be maybe a gene editing approach, but actually we as a company are considering also other type of approaches because the gene editing isn't the only one, and it really depends, like John has been pointing out, it really depends what your target is. And there is a bit more to think from that point of view. So it's not just it's not just gene editing.
Well, on that note, I mean, let's look back just for a moment from 1953, since we understood the structure of DNA. 2004, we completely sequenced one whole human genome, and now with kind of the sequencing technologies, machine learning, AI coming in, the amount of data and information and understanding that we're getting is kind of accelerating. So what's next in a way, Debora? Okay, gene editing aside.
Yeah. So we everybody talks about this, right? So what's AI and what's going to happen and what are people eventually going to do? What we generally believe is that so first of all, there is a lot of data, so exactly like you said. But where is this data collected on? And we all go around and say we have the right platform and we all have to recollect, data on a specific cohort, on specific type of biomarkers and so on and so forth. However, where I think everyone now is heading to is to rationalize a little bit all this type of information and make it, commercialize it somehow. So defining patterns, trying to find, methods, and really they need to become from everybody says AI, machine learning, from a computational approach. So something that is able, an algorithm that is able to process data and to find patterns and then to connect the different
electronic hardware components for the data storage, which is really energy consuming. By 2025, more than 1/5 of the Earth's power will be used for operation of these data centers.
He's chairing tonight from Mumbai.
That's not me.
Aha, yes. Okay, so yes, yeah. So what I was trying to say was that basically, so one way to go is lots of us at the moment are trying to find the patterns, elaborates on larger and larger data sets, almost population-based data sets, so that we can extract patterns and we can find, some ways forward. We had a chat this afternoon, Prashant, and I also think that where the technologies are moving is also start thinking, how can we intervene a little bit faster rather than spending 15 years to do a discovery, because that's the time that it takes to take a drug to the market and is very expensive, as you know. I think by using these technologies and these large data sets, there is an opportunity to reduce that initial discovery step. And this is something that Enhanced Genomics is trying to demonstrate. So what we are trying to show is that if you're able to identify from the very beginning of this story, the right gene in the right cell type, you already are a long way towards your therapeutic discovery to understand, what your real target is going to be. And therefore you can shorten a bit that discovery pathways actually. But from a data perspective, we generally believe we are building what we call an artificial labs of the future. And I'm sure a lot of us actually feel like that actually. So
Louwai, I mean, you would relate to a lot of this with the cost of sequencing, slashing, you're able to kind of relate and get the data that you need to make better predictions. I mean, that's exactly, you've got some pretty grand ambitions for your company of what you want to be able to achieve in terms of, exactly as Debora says, getting therapeutics to market more quickly, more efficiently?
Yeah, I think it's a really important point. I think this is why AI and genomics go together so well. So the way that we're trained as physicians is based on this Oslerian paradigm from William Osler, who was born in 1849, where we define diseases based on a top-down approach. So we go from symptomatology and organ systems, and we draw circles around things, and then we say this is X disease, and we try and come up with treatments for that. And we look at the molecular features nowadays sort of backwards. But actually, biology doesn't work that way. That's a human construct, all of that. That's for us to, as clinicians or as a society, to make something rational and make something that we can do something about. We can try and understand, make diagnoses, try and come up with therapies that way. But it's not biology. And so what's happening now with the ability to get that kind of data, where we can sequence a genome in hours rather than in decades, and for £100 rather than a few billion. We've now got all these, like Debora said, vast amounts of data. It's no longer human comprehensible, even though this is much more accurate and more representative of biology. Human ways of thinking and processing aren't going to be relevant anymore for that. So what is now really exciting is the fact that that type of technology is becoming mature and coming of age at the same time that the computational technology is also coming of age to address that type of problem. If we had all that data 50 years ago, this is completely useless. We'd never been able to process it. And if we had the technology with the computational abilities we have now, without the raw data, the model's only as good as the raw data that goes in. Yeah. Now we have single cell sequencing, state-of-the-art CRISPR tools. We can incorporate all of those. So they're merging together in a very unique timing, and that's why I think this field is going to be very big in the future. And this is the right time for this area now. And
yeah, I've just got a question that's come in on the AI part of it. Are you kind of building your own code, your own algorithms, or are you able to kind of use some off-the-shelf AI tools to kind of process and support you with your kind of analysis?
Can I answer one thing first? Yeah. I think we had this conversation in a previous forum. Actually, there's a long discussion about AI, machine learning, and all this definition. Actually, so we are building an AI company, just to be clear. And I had this conversation internally with the VP of bioinformatics and so on and so forth. So. They all, first of all, like to call themselves data scientists. So, because like Louwai just said, actually, so there is this huge amount of data, and there is also some very advanced technology that they need to be actually married together so that they can operate. And like he said, actually, you need to think of a code, of an algorithm, or a concept that basically allows then the machine to read all this data and to then rationalize it in a certain manner, actually. And so that's all it comes down to. Because obviously, as human, we wouldn't be able to see this across these huge data sets. And what my team constantly said is that it is really, really important that you trying to understand what it is the question and what it is that you're trying to extract out of these data sets, actually. So you need to be very clear in your mind of what it is that you are query, actually. Otherwise, the answer is basically as good as the input, realistically. So or what you are asking or what you're putting in and what you're getting out is exactly the same, actually. They correlate to each other. And so I think that this is an important point, because everybody likes to say machine learning and so on and so forth. From our point of view, just to say that we are collecting data because we have a platform that now allows us to have another dimension. But at the same time, like Louwai said, we also use sequencing data, we use other type of expression data, and we have to train our machines. So what they need to do is to read each data set, trying to extract a certain type of information until they actually learn a specific pattern. For instance, we take a specific cohort of people who have developed a disease, and then we take call it a matching cohort of people that is healthy. So then we have collected similar data sets for both of them, and then the machines go and read all this data, and then they basically extract the patterns or the differences between the two of them. Louwai, maybe you want to explain this from more a medical point of view, but that's...
Yeah, so I mean,
Yeah.
the question was, are we building our own algorithms or using previous ones? I mean, all of science is built on the shoulder of giants. So we're taking things that have been developed before, but we are developing our own novel, unique approach. So the way that we are doing this, and the type, we're writing our own code to do it all. So it's all sort of it's a very building on previous approaches, and it's, as Deborah sort of hinted at, it's really understanding the problem. You have to know what question to ask. So you might be able to apply an ensemble learning technology and technique that's not novel, but no one's ever thought to ask the question in the way that you've asked it. And that really goes as well for the future of this field. It's not only, so I hate saying machine learning and AI, even though that is what we do, because everyone kind of says it, even if it's not really kind of as much of a big part of what they're doing. But what we're now progressing towards, and this is very exciting, is a lot of what these technologies are trying to solve are learning questions. So for example, a feature selection question. So a lot of what we do is around feature selection and prioritization. So if you imagine an algorithm that recognizes faces, you put into the model 10 million faces, and it extracts what the principal components are to recognize that face and distinguish it from somebody else's face. And when you do this in medicine, you can get these algorithms where or these technologies where it's looking at, say, a brain MRI scan or a histology, and it can make the diagnosis correctly better than the human can. And when we look back retrospectively and ask, how did it know this? We actually can't see it. Even with the retrospectoscope, we don't know what it is the AI is using to predict that this person has got this disease. And so it's extracting principal components that as humans, we're just not able to recognize. And that's exciting because there's another technology that's now coming of age. And this, again, I don't like saying it because I think lots of companies use it as a bit of hype and it's not real, but in our case, it's actually critical to what we're doing, is quantum technologies. So quantum computing is actually not superior to classical computing in almost any application at the moment. It may well be in the future, but right now. But what is useful is a particular use case, which is applying quantum annealing technologies to feature selection problems. So for example, with us, our input for our model will extract about 70,000 features that will predict what makes cancer cells survive and not survive. And on our current high performance computer that we're using, it's a sort of a massive supercomputer effectively, it takes about 52 days to run that analysis. But with a quantum computer, and that's sort of quantum annealing technologies have been shown and used in this use case. So D-Wave, for example, has a quantum computer that can do this, can solve that same problem that would take us 52 days, can solve it in 10 minutes. And so that's useful for us because we're building it into our platform right from day one. So it's in the fabric of the actual engine itself. Because when in the glioblastoma, it will take 52 days, but in our next engine, our next kind of input data functional genomics screen that we put into that, it may take 2 decades for a classical high performance supercomputer to solve that problem. But the fact we've got this quantum technology incorporated from day one means we can solve it in days to weeks instead of decades. And it changes, again, it's a step change in what we can do. And it's all happening at the same time. The classical computing technologies, the biology and the CRISPR editing technologies and the quantum field all coming together at the right time. So that's why it's a really, really exciting future that's coming, I think.
John, do you think this kind of a correlation between different fields and different data sets and analysis and data at the cellular level, understanding the genotype, do you think this kind of crossover and information from different areas is the way forward, and how do you think it would actually come together? I mean, a lot of people are kind of analyzing different biomarkers and correlating it to kind of the different kind of disease progression. But if we were to pull all that data from everybody together, would that not be the way to go forward?
So I'm going to say things are some things are cliched and some things are romantic. I'm going to say here. So first of all, it's very hard to make predictions, especially about the future. That's the cliche. The romantic thing is on the nature of artificial intelligence and machine learning. So we've heard from Louwai, about the rigorous training, doctors undergo in diagnosis, fitting things into sets, what's in, what's out. does this disease fit this? Yeah, which is a human construct. And a lot of the academic learning we've all gone through to various levels of success has been in that formal structure. But there's other sorts of human intelligence. Okay, so if you're playing football, you're going down the wing and you're trying to cross to someone in the far post, you know, you're having to, there's no way you're solving those equations of mechanics, you know, to work out, you know, what angle to kick the ball at what velocity in three-dimensional space with your wrong foot. You do it because you practiced. And so when we set up a computer with the training set, it's doing much the same. So it's working in this way, which isn't that unfamiliar to humans, but is distinct from formal knowledge and a formal algorithm of stepwise steps. Of course, stepwise steps are, you know, stepwise algorithms are how these things are constructed. But how, you know, how, if you ask someone, how do I do that? You know, it's like, how do you read someone's lying to you? How do you decide whether to like one? It's a different sort of intelligence and thinking to the formal stuff. And it's something we share with other animals as well. So artificial intelligence, if you like, isn't that artificial, but what it does have is a very good memory. So drug discovery is getting harder. There's Moore's law, which we hear a lot about. The Ehrum's law, which is the old sort of cliche from Nature Reviews Drug Discovery, about how everything is getting harder. This technology will not stop that, but it's essential to make any progress. Drug discovery, you know, is always the search for what hasn't been done before. Generally speaking, the easy things have been done, either because they were technically easy or conceptually easy to discover. We're just left with harder and harder problems. Occasionally, you see an opportunity which, you know, is a confluence of knowledge and surprise, where you think, oh, existing technologies can make a big impact fast. But more often, with these sort of target ID technologies, you're getting a better level of understanding, or someone or some system has a level of understanding, but it's not the only way to access the knowledge about whether something you're trying is an effective route forward or not. So with these questions, it's always an issue of how difficult is it to do an empirical experiment versus build a conceptual network of what might be going on in a cell, in a person, patient, or whatever. Where do I think it's going? I think it's really hard to say. I'm always skeptical about breakthroughs. The reality usually surprises me about how positive it goes. Clearly, these are exciting technologies. They can do things that, you know, no committee and no person can do because they can't hold all the information in their head. I think some of the outputs of machine learning are intuitive, but when it comes to describe it in the academic press, whatever fine detail and general sort of holistic view the system has is often collapsed back into a story which is mapping on to, you know, a stepwise narrative suitable for an academic paper. So I think there's a difference between how it's done in reality and how it's presented. I think drug discovery will remain hard in many aspects. We will be able to do more. And the good thing about the patent system, rather than the copyright system, is that after 12 years of unaffordability, things do become you know, affordable for society, and the innovators of today can go on to solve new problems.
So on that note, I mean, just to kind of, as you mentioned, there's a lot of exciting technology, there's a lot of potential. So I just, as entrepreneurs, as you all are, and in John's case, you know, at least 3-level, 4-level entrepreneur in parallel, how are you finding the funding world? I mean, is there a lot of capital now chasing your ideas? How difficult are you finding putting your ideas into practice and getting the money that you need to put it into practice? Louwai?
So I think, again, now is a good time in that field. There's a lot of interest in biotechnology. I think a lot of the people who've had a lot of success in tech, for example, we straddle the intersection between tech and biotech. So we're getting interest in being able to see the difference in not just wallets, but also culture from the tech side and the biotech side. So a lot of people are recognizing that now. And I think also there's a move in the academic sphere towards more close partnerships with industry. Because I think ultimately what's driving a lot of scientific progression is, have you got enough funds to develop it? There's sometimes a bit of a culture, if you throw $100 million at something, you'll figure it out. But that's not quite true. But there is definitely an element of the more resources you commit to something. There is, I mean, for example, looking at cancers, there's almost a linear correlation between the amount of money that's been spent to try and cure a cancer and its prognosis. So breast cancers used to be completely a death sentence, and now they're very treatable. Brain cancers, because they're rarer and very far less money has been spent in that area of research, is still a completely lethal disease. So I think the academic sphere, especially with Brexit and the drying up of academic resources, are recognizing that the best way to get therapies to patients now is to think about it more in an industry type perspective. How can I partner with industry and the pharma companies that are going to eventually turn my bench side academic research into something that's going to help a patient at the bedside? So I think there is a bit of movement in that direction. And I think now is, again, a good opportunity for raising funds in this type of sphere.
Are you feeling that as well? I mean, you're on both sides of the system as an investor, as part of an investment group, but also getting some of your ideas funded as chair and CEO. John, are you seeing a lot of potential from your ideas in that it's getting recognized by the venture capital community?
Yeah, so you might want to bring your microphone up, Prashant.
Yeah, thank you.
What I will say is that it varies on the opportunity. I think there's more money in the system than there's ever been, which is obviously good. You know, there's still a lot of fashion, and some things are hot and some things are not. So with any opportunity, there's a time, you know, that it's ripe, and there are fundable and unfundable teams trying to do it. And it's always difficult to get sensible amounts of money because of the level of risk and the level of trust that has to be involved. So certainly there is new money coming into biotech from the tech side, as Louwai said. And yeah, they have, even in the UK now, a nice example in Accenture of how it can make money, and perhaps in case of Benevolent, a case how it can raise money on huge valuations, but we'll see if, you know, if that's sustainable. So with these sort of three things I'm involved with Epitopia, it was relatively easy to get £10 million to go to start it. And that was partly because the university had done some legwork. There was human factors, like Canadian investors wanted to invest in a Canadian company, but there was no one in Canada to do it. And so we created this sort of sister company structure to bring in UK investors, Canada investors. So it's all sort of path dependent, history specific, and driven by political factors. But we were able to differentiate ourselves from many other companies in the space by our particular ambition to make a really big difference, you know, rather than extend life by a year, but by 10 years, and a credible case that in some occasions that could be done. And then also an effort to put into the pitch that it wasn't all or nothing, and we have fallback positions of more, you know, conventional approaches in, you know, stepwise iterative cancer therapies. With the other company I'm running in remyelination, there's a lot less competition, which is good. You know, there's probably as many buyers as there are companies in the space, but there's obviously been less success in the field, and therefore there's a lot more skeptics that it can be done at all. And then there's a question of, with any investment question, it is there an exit possible, a liquidity event for any financial investor to return their money at, you know, a significant multiple at the timelines they need to. And certain business models and certain size of company are in the sweet spot for certain companies. And with Pheno, I think we'd have to look quite hard to find them, and that's why it needs full-time management. Finally, with Pencil, we've taken tiny amounts of equity. You know, the investment we had from OTH, which was, you know, 5 figures, you know, is in the, you know, 7 to 12% of the equity gain. We've taken a convertible loan to avoid a valuation decision, because the future fund was there for a few weeks, so it's a good time to force the hand. And now we've got a technology that works, And the interesting thing about this, there was no academic precedent. It was a pure invention by the founder, and there was no third-party sort of leverage data you could say this is going to work. So it lives in the CRISPR world, so people know it's a valuable technology if you can get it to work, but it lives in a competitive world because they're a long way ahead. And what I have to do there is to find a business plan which brings in enough cash on terms that the existing investors and the new investors are both happy with, which isn't just, let's spend 200 million and see what works, because we don't have a team back with FRSs, and successful exits, but that sort of approach would work. So we have to come in with a sensible plan of, we're going to do this, 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 and this. We need 10 million to get, this point. These are our fallback positions, and this is why we should do this and not the other. And when we get that right, because gene editing is extremely cool, we will have money, no problem. But we have to think carefully about what it is. So I think any business plan, needs to be decent to be funded, but in the current environment is a lot more permissive. And so the good business plans tend to get funded rather than just 10% of the good business plans.
Okay, Debora, I've just got a couple of questions for you. Firstly, if you could run into the funding question, but first is a question in from our S-Menon. And how do you feel about the quality of genomics data available to you for AI analysis? And do you have to end up generating your own data? And then you could come on to the funding question.
So I think there are quite a lot of good data sets available. Some are publicly available and some are more private. The quality is quite good, and there are different technology that you can use to date. I mean, even, not just Illumina, but also there are other platforms like PacBio, and even some of them, Oxford Nanopore is getting very close now. So depending on what kind of, or what part of the genome you are interested in, you can definitely get some good quality data set. And I can see that possibly also the next question is for me. We do generate our own data simply because we, not because we want to reinvent the wheel or create a new genomic sequencing, simply because we are looking at 3D interactions that they could not be detected with the current platforms. So hopefully in time we'll be able to develop something that allows to do sequencing as well as looking in the 3D at the same time. So reducing the number of data sets that people need to collect. But at the moment, we do use some of the existing technology and there are good data. Genomic England is a very good example. They have very good data sets to use. In terms of funding, I was also thinking if I could say something that maybe hasn't been already covered by John and Louwai. And one thing that I was thinking, going back again to the topic of today, which is genomics and AI, is that from a funding perspective, there is a lot of investment and a lot of interest at the moment from the investors in the genomic space. And this is because Illumina, one of the biggest players in the market, Illumina's patent actually have expired last year because there have been very huge transactions. Maybe I don't know if people have noticed, but there was this rebuying obviously of Grail into Illumina. This transaction is still going, but there was also this big acquisition of Exacton, based genomics, and there is Freenome and a few other genomics companies. They're doing huge fundraisers actually. So there is a lot of interest from an investment point of view in a genomics company. Now, either those because they use publicly available data set, and there are a lot of companies that they do use publicly available data set, or because we are producing our own data set, the interest is there. And the reason for that is because I think mainly, or at least for the company I'm involved with and in the past have been, is because there is this big promises of this new platform like AI and this new discovery coming up and allow you maybe to do drug discovery, like John said, with different eyes and in different ways, because it is becoming more and more difficult, and what everybody is after is actually new targets, new real targets that people could work on. So that's to answer the funding on genomics. The other thing that I think I noticed, it seems to become a trend, is that the Series A are getting bigger. So particularly in the last 3 or 4 years, I mean, there have always been some people, either because very successful entrepreneur or because they had an amazing incubation type and could generate enough data. But in general, it looks to me that the Series A are getting bigger from the beginning. So rather than doing that stepwise approach where people, pull up the first million and then maybe 8, 10, and then so on, it looks like the people go straight into 10 million seeds fund, if not 20, and then the Series A is going to be, I don't know, 50 or 80 or something like that. So I think that's a trend that I have personally noticed. And I also would like to make a remark, Prashant, so and then I'll leave it here. From my experience, there have been fields, and maybe Louwai can comment on this, like, the epidemiology space, like infection disease, has been a really dormant field, and there wasn't that much going on up until 2 years ago when COVID popped up. And now all this technology on vaccines and all sorts of, related platforms seem to have become, again, the flare of demands. And like John said, it really depends on the time. You may be on the right time. And I stop here because I know I'm using a lot of time.
Well, there's one observation I'll make about kind of the amount of money that's going in. I mean, look, AI definitely kind of narrows your shots on goal. It gives you better predictions of where you want to kind of start your drug discovery program, essentially. But the cost of then doing drug discovery, that's still quite a capital intensive effort required. But look, and the kind of issue of funding and the point you made about Series A funding, I think, Debora, is quite interesting, because I'm interested on your views, because genomics and even AI and technology, Cambridge, it's a strength for the UK, but both those kind of merging fields, Cambridge and the UK. In those fields, Cambridge and the UK has probably had an edge, since for the last 30, 40, 50 years. Do you think we still have an edge in our ecosystem? And what do you think the future holds for our ecosystem? What do we need to do to sustain our edge? Yeah. So that question to any of the three of you, whoever wants to come in, really.
Yeah. In terms to sustain, I think maybe, we can, I'm sure everyone will have a different ideas. I do think that Cambridge and the UK in general is still very big into the genomics, apart from the fact from the many reviews that possibly everybody may have read, from Genomic England and the government in itself that has invested in this very large sequencing projects, actually, and that they're constantly campaigning on that. So I think Cambridge is still doing very well. There are loads of companies. We Enhanced Genomics are based in Cambridge, but there is a huge ecosystem in the genomic space, and trying to address different, different type of therapeutics. It could be cells, it could be, drugs discovery, and so on and so forth. One thing I thought about our conversation was this, actually, that one of the investors have said to me when we were talking about investment type, said, we like the genomics investment, because if you think of any investment, and let's say the genomic investment is 10, to them, then the next investment will be maybe the diagnostic, which could be 100, and then there could be then the next one that could be drug discovery, which is, another 0. Basically, the reason why investors seem to like this genomic space is, though, like you said, there are still quite a huge resources that need to go into it. From an investment opportunity or point of view, it still looks smaller, and possibly more attractive. However, the closer you get to the value chain, to the end product, possibly also the bigger is the jackpot. So, a lot of us still have to demonstrate, that therapeutics huge, jackpot. But that's what I think, actually.
And Louwai, you've got some pretty interesting views on how, we compare globally to, even the American ecosystems. If we club, You had to look at London, Cambridge, and Oxford together. You were.
Yeah, so I mean, I spent a while in Boston in the biotech sort of center of the world, and there's no question Britain is not as big as America, but America doesn't work like that. So Britain has got a unique setup where we've got one single system. So for example, Genomics England works because the NHS is a single unit, and that's significantly bigger than Boston. All of the kind of patient data you can get from that resource. And if you look at the infrastructure, the type of academic professors, the quality of the science, and what we've done with the limited resources we have, if you combine, say, Cambridge, Oxford, and London together, and that is bigger than Boston. But what it's lacking right now is a single kind of managerial or management approach where somebody who's able to exploit all of those things at the same time, bring that together and package it as a unit. And when you can combine it in that way and have management and overseeing all of that potential, then you can really not just compete on the world stage, but actually outperform them. And we tend to do it a lot here. We have, if you look at the number of Nobel Prize winners, the quality of the science that comes out of this part of the world, America does, we're on a par with them, despite the fact they have significantly more funds, which goes against the idea if you just throw $100 million at something, it will work. Well, what happens if you throw $100 million at something and you have the right people and the right science behind it as well? You should do better. So there's a lot of exciting things that you can, I think the potential here, particularly after now with all the problems that are coming up with academic funding and the way that COVID has actually allowed people to see across the borders. So it's much easier. We have meetings with American investors that probably would have to fly over there to sort of speak to them. Now I can just do it on a Zoom call from my flat. And that's the sort of potential. People are more comfortable having these kinds of meetings and taking bets on companies farther apart. So I think if you can have a setup where you've got the British ecosystem, but with the American funds, which is where the world seems to be going, then there's a lot of opportunities that will come out of that.
And before I kind of roll on to John for his comment on this, as you're the most kind of recent probably entrepreneur of the three, we've got a question come in. As a PhD student who has an idea, from where can I start and how can I get data and get cracking on the funding?
That's a great.
Would you like to give your insight or suggestion to him?
Yeah, so it's good. There's two bits to that, I think. So first of all, it's the passion side of it is important, because the ideas, although ideas are key, what happens a lot with entrepreneurship is you pivot a lot, the ideas change. Ideas aren't as important as the team driving it to start off with. There's a classic kind of cliched statement from investors that investors will always back a great team with a mediocre idea versus a great idea with a mediocre team. Just because what you think you're going to do now will not be what you end up doing. You'll start doing things will go wrong. You'll realize that actually the value on what you thought you were going to do isn't quite as good, but you've discovered this other really great value that's kind of linked. And as you work on it, it's very similar to academia, actually. I think there are the two sides of the same coin. So in academia, it's a comforting thing. You're used to science going wrong. You weren't expecting it to go right the first time. You're expecting to pivot and have to figure out how to solve this ridiculous thing you never thought was going to be a problem. And that's exactly how entrepreneurship works. So from the idea point, there is an element of commitment to it that I'm going to see this through. Getting the right team around you. So for me, I've been looking for co-founder, like Lisi, for example, my co-founder, we work really well together. And I've had sort of other kind of people you need to find people, you can't do it all on your own. So you need to get a skill set of a team that has complementary skills to yours that you shouldn't all be able to do the same thing. Otherwise there's no point in the team. So getting that team in place, and then there's always going to be the strategy aspect. So there's a killer proof of concept experiment you're going to have to be able to show. And if you can bring down that kind of cost to it being really the minimal possible cost that you can get it to, and demonstrate that proof of concept, and make sure that it's not a proof of concept that you think is important. You've got to figure out what are the value inflection points for others. What is it that if I went to an investor now and said, I've got this great idea, here's the data, are they just going to turn around and say, I don't care about that data, I want to see this other data? Don't do that. Figure out what is the data that they want to see first and tailor everything you do, work backwards. So the way I sort of described it, I started with the patient. I want to get a drug that works to treat that patient. How does a drug get there? Went to the pharma companies. What does the pharma company want to see before they acquire a drug? Then got to that stage. They went to the VCs to say, what would you want to see to put in the money to get me to a thing that would get it to a pharma company, that would get it to a patient? And I work backwards from there. So it's really about that kind of way of thinking. So I'd say the summary of all of that is the team, the strategy, and the passion behind it. Those three things together is where you'd start from a PhD student with an idea.
Oh, that's a fantastic answer, Louwai, in there. I could have done better myself. Brilliant. And John, your thoughts on the on where we stand in the UK, Cambridge, in terms of our skills, our know-how on the world stage?
Yeah, I can give a very long answer to that across many domains. First of all, my advice to the PhD student with an idea is make sure the idea is very ambitious, and that, you know, a small investment, as Louwai said, can have a big impact. Then, I mean, I think it's hard, very hard, to raise significant funds as a young entrepreneur in life sciences. Much easier in tech. The team is important. It can be done. It is very difficult. Don't worry if you can't, because there'll be a chance down the line. So I don't have, you know, the perfect Oxbridge medic CV that some people do. You know, I sort of bounce along in mediocrity. Now, I've worked with Nobel Prize winners and got a science paper and put that lab back on the map. You know, you could say I'm a slow developer, but the reason I was selected by Advent was an honesty of aspiration, of wanting to make a difference, and, you know, being prepared to stick at it, and not necessarily blindly and stubbornly, but to, you know, pivot as necessary to make the overall mission work. And that there was a team, you know, an ability to put a team together. You know, and it is a team that functions, but someone has to assemble. There's more. I mean, even leader type people still want to follow sometimes, and they want to know that, you know, that it's not just them thinking it's a reasonable idea, but other people as well. So getting company off the ground is difficult and takes time, and it's a persuasion. And you've got to listen to people saying, well, I wouldn't do it like that, because they're probably right. Now, when it comes to the UK environment and Cambridge in particular, so clearly there's a great history of innovation and basic science here, and there's a great history of cutting edge products as well. So where the system differs from the US is at multiple levels. First of all, there's a longer history of life science venture capital, and at scale, going back to Genentech and Biogen and et cetera, from the late 70s onwards. Second, there is a functional public market for life science companies, and some of the US ones have remained independent and have become huge $50 billion companies like Gilead. I'm amazed they haven't changed their name yet, because ISIS did, didn't they? But there's you know, there's a history of companies making it big there. I think there's more ambition from investors as well, and more ambition from management teams. When it comes to Boston in particular, this is VCs being lazy, and, you know, they sit in their office in Newbury Street, their Third Rock. You know, people come in, pitch, and they can always back, you know, another idea by Harvard Lodish, you know, for his 17th company. You know, and there's be top people come along and say, my postdoc's got this idea, let's set up a company. And they usually give those companies quite a bit of money to play about with, and not all at once. So an announced Series A of 125 million, that company did start 4 years ago with $1 million, and it's built its way up just under the radar. But what they do have is a lot more money to put at it, and they have a lot more management teams which have done it and had an exit. And the most important things for the US life sciences industry are the ridiculous price that American, you know, consumers, for their insurance agencies and Medicare, pay for medicines. And that just attracts money. The pharma companies are all there, even if they started elsewhere. They're all in Boston. They used to all be in New Jersey. They're all in Boston now. There's tons and tons of money in the system. For an innovative medicine, 65% of the revenue and probably 100% of the profits will come from the US market solely for a long time. And, you know, clearly it's got crippling costs for America, but it isn't going away, because I think the legislators are in the pockets of various lobbyists. So America will have this sustainable sort of lead on company formation, because there's a greater potential for company exit. The ecosystem of takeouts at the big multi-billion-dollar level is there. And in America, people will say no to a $2 billion offer, but not many people would do here. And so one of the issues Britain has, together with Europe, is there isn't that ecosystem of analysts, of pension funds, of investors investing specifically in European biotech companies with the idea that they become big and the pharma companies of the future. Everybody's looking for an exit too early, I think, for that really sustainable things. If you think of the companies we could have had that could have been great, like CAT, which was involved in the production of the world's best-selling drug. You know, they sold out for less than $1 billion. I know it was a long time ago. And I believe AstraZeneca.
Yeah, this is definitely a conversation, you know, for a whole nother Enterprise Tuesday. This one, a very good one though, and very important.
But we have our strengths, yes.
We definitely do. And on that note, I mean, a question is, we're going to ask you for your closing comments in just a moment, but this a question has just come in for an international student and said, well, should they do their biotech startup back at home in their home country, or should they come to Cambridge? My personal view, I want to chuck my own view in here as well, that you should definitely come to Cambridge. It's a great cluster, it's a great community, and having those face-to-face interactions and participating in that community along with these fine ladies and gentlemen that you have here is what can really help you. It's a huge team effort. Although I think Zoom is lovely, there's no substitute for kind of being in and around all the actors that you need to kind of help you get your idea off the ground. I mean, this is my personal view. I don't know, did you have anything? Would you agree with that, team panel?
I would like to add one thing, that Prashant, very modest tonight here, but in answer to both the PhD student question, Prashant and his brother are some of the people you should contact if you're thinking of a business venture, because they are some of the very well established and very experienced people that they actually will be helping you to build a company if there is some legs there. Actually, I just wanted to add that.
Well, thank you for the plug. We always appreciate a plug for yeah, we love doing it. We love getting in there early with seed stage biotech companies, doing everything we can to kind of support that. It's a passion of ours. Thank you. Let me run into kind of your summary comments, really. I think I'll give you a wrap up, but is there anything you missed that you wanted to get out there or share, or what are your closing thoughts on kind of everything that we've covered today? And yeah, would you want to go first, Debora?
Okay, yes. So my closing thought are this, and I also was rereading again the text for today's presentation. So I think the UK is a great place to do genomics. I think that it is still one of the forefront from a technology, from a team perspective, and also from an investment point of view. Cambridge, we are based in Cambridge. We really think it's still the place is very thriving, and there is a lot of cooperation, and it's not just for genomic. As Prashant alluded before, there is a big team and a lot of people to work with. If you want to start your own company, also to answer to the various people in the audience, there is plenty of people you should be talking to, and but trying to get your ideas together, like Louwai said before, because there will always be somebody ready to back you up, but you need to be prepared and able to explain what you want to do and where you want to go. Those are my thoughts for today.
Thank you. Your quick closing thought, John?
I think with respect to starting a biotech company in Cambridge versus another area, I think, you know, unless it's near market, so to speak, and revenue journey, you have to talk to specialists who have the patience and know how to keep the cash coming in to a company where expenditure is greater than the revenue. There's a skill in that. There's a skill in selling the narrative, and you know, it's very, very difficult to come up with, you know, completely new story to investors and get significant money, because, you know, it will be just too hard to pull off. I think, you know, once one's established, you know, having, you know, a center in another country is potentially an interesting differentiator. And you can say, oh, it brings in different cultures, blah, 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 blah. So I think you can end up with a foot in two camps. I think, you know, Prashant is probably someone who's played that game very, very well. What I'd say about genomics, I've got nothing additional to what I said. It isn't it's a trendy area, but it's an exciting area. Maybe it won't have the impact of the most hyped, you know, iterations of boosterism on it, but I think it is a significant impact. These new technologies will lead to new breakthroughs in medicine at some kinetics or other, and it'll be fun and interesting and worthwhile to be involved with that. So I do recommend, life sciences, industrial life sciences as a career. It's been fantastic to me. People should definitely, if it's something they're considering, I would recommend it as an aspiration and a dream to continue.
And over to you, Louwai.
I guess I'll just answer some of the questions that I can see still. So in terms of the international question, I think serendipity is not something to be understated. So being in the right place does increase the number of serendipitous interactions. The whole of the Boston ecosystem is set up to be, you have the VCs, the entrepreneurs, the tech culture, the pharma companies, all in a tiny little town having dinner with each other all the time, and conversations happen and luck happens. But it's not really luck, you're actually just in the right place at the right time. So being not necessarily Cambridge is a great place, but one of those hubs at the early stage will make your journey a lot easier. It's significantly easier to build a biotech company in Cambridge than it would be in a place that doesn't have that. So I think I agree with the idea of maybe later on moving down when you're established, but for the early days, starting with that. And then just quickly for the other two questions I can see, because it's quite relevant. So in terms of the quality of the data for genomics and AI analysis, it's always going to be your data is only as good as the input data. Your model is only as good as the input data. So we're generating our own proprietary data for that reason. And we don't try and overcome problems. We remove them from the input. So looking at what companies are doing, looking at what kind of input data is just as important as looking at the kind of models. And the last bit about predictive, whether physicians will take up these kind of methods without rational kind of wet lab validations, that's not what we're talking about. The AI is to predict, and then you go and confirm biologically. It's always going to be that way. You're always going to have your in vivo experiments. You're always going to have your clinical trials. The beauty of clinical trials is it's completely agnostic and doesn't care about how your drug works. It's just answering the question, does the drug work or not? Most drugs I prescribe, there's some little bit of biology that, it binds this receptor. How it actually works, I don't know how paracetamol works. No one does. We know that it binds certain things, but why does it get rid of your migraine? It's a mystery. All we know is in clinical trials, I can prove that. So the AI is there to rationalize that, narrow down your search space to make it something instead of a billion combinations, just like 5, and then figure out and test those 5. It will never be one without the other. AI does not replace, it will always go down to clinical trials ultimately.
Great, thank you. On that note, I'd like to thank all three of you. I mean, it's been a terrific learning experience for me, getting to know your stories, your research areas, connecting it all together, going deep into the science, then coming out and looking at the cutting edge gene therapies, the current uses, future applications, looking at the history of genomics, relating it back to Cambridge, looking at the regulatory, the ethics, the entrepreneurship, the funding, and what a tour it's been over the last kind of an hour and a half of the world of genomics and AI. So thank you very much once again for making the time to attend, and for your really insightful and candid responses to the questions in the discussion. Thank you. Over to you.